Thank you so much for having me. And I'm really sorry I wasn't able to make it. It turned out to be too much at the very beginning of the semester, but I've been listening into the talks and really enjoying being there, at least for the talks in your afternoon. And actually, I remember here saying this yesterday, that listening to all the talks makes you want to rewrite your own talk. And I was certainly having that experience that um, this is a different audience than or a different set of talks in a different environment than I'm used to. So I'm really happy to try to talk about how the material I'm going to present connects to the previous talks and the other issues. It's not trivial. Anyway, but it's really interesting to listen to all of these other talks that have focused so much on cross-species comparisons, on brain evolution, and on connectivity, because I am not going to say anything directly about cross-species comparisons, about brain evolution, or about connectivity. But I think actually that some of the deeper issues about where the mind comes from are really similar. So anyway, I welcome a chance to pull out those connections. Great. So anyway, can just get started. So for me, this talk also begins with a gap between disciplines, but it's a slightly different set of disciplines than the ones we've mostly been dealing with so far. And actually, it's interesting that a number of the questions over this workshop have been about how brain structure relates to function. Because for me, I always start with function. So I came to the question about brain development actually originally from being a researcher on infant social cognition and social behavior. So I started studying development behaviorally in the field of social cognitive neuroscience. And in that field, for the last few decades, there's been the kind of collective development of a very rich body of knowledge about the infant mind, how infants see their world, how they think. And that very rich field, whole body of knowledge, has been developed using mainly behavioral measures of infant looking, right? So where do infants look when you show them a little play? And then to some degree, Bayesian cognitive models of what kinds of processes in the mind could lead to those looking behaviors. And so from that field, actually, could I see a show of hands? How many people here feel familiar with behavioral measures of human infant social cognition? Is this a familiar field to people in the room? I see 3 hands. Okay, great. So not so familiar. Okay, so for me, this is background knowledge, but please feel free to ask me about this field because this is the thing I actually do know. I will show you one example from my lab, but I'm happy to talk about much more about where this idea comes from. But just to start with the biggest possible picture, I will say that the field has come to a view, or my lab and many collaborators and other researchers have come to the idea that from very early on in infancy, really young preverbal infants understand the world in terms of very abstract ideas about agents and goals and knowledge and relationships. And the infant's knowledge is derived from rational inference from sparse data, both through passive observation and through goal-directed active exploration. I'm not talking about walking around at 14 months. I'm talking about actively choosing who to look at and when to smile at 3 and 4 months, right? So very young babies, before they can do almost anything, are already understanding the social world in terms of very rich abstract meanings that they get through both observation and active exploration. Okay, so if that's not a familiar claim, I will show you some of the kinds of evidence people use, but again, very happy to talk about it. And for me, the question when I came to the brain is, okay, so how is it? What is it about the human infant's brain that supports these incredibly rich social and cognitive abilities from very early on in infancy? But the field of people who study human infant brains in developmental cognitive neuroscience has tended to have a very different framework theory of infant development, has seen infant brains and minds from a very different perspective. So this field, which is closer to the kinds of fields we've seen in this workshop, uses direct measurements of neural activity in human infants, and in this group, much more in comparative studies, but in my field, more in human infants. and also uses models of networks, models of connectivity and connectivity change. And that field has almost the opposite idea about how function might emerge in infant minds, right? So the idea is that babies might start with very simple, concrete senses, right? The early vision, early sensation, and through gradual association and self-organization, which is imagined to function mainly through model-free reinforcement, there's kind of gradual growth of a very simple cognition in infancy that could slowly, and people in developmental cognitive neuroscience are often measuring changes over decades, right, in brain structure and function, lead to kind of adult cognition. So for me, this is a very interesting tension, right? That the people who study the mind, the infant mind, think of it as capable of incredibly rich, abstract, rational, goal-directed exploration from age 2 or 3 months. The people who study the brain are suggesting that higher order cognition takes decades to develop. So how can we connect these two approaches? And in particular, again, for me to ask, what is it about brain development and the way human brains are designed that lets us have the minds that we have, and that lets infants develop the minds that they have? So for me, in my current research, I'm interested in trying to force these two fields into contact with one another to make predictions for the same phenomena, because hopefully that's a way that we can try to understand where they're each coming from and how to change them. Okay, so today I'm going to talk about a specific case study where I've tried to argue that these two fields offer different perspectives and can be made to make different predictions. And that is specifically the origin of face-selective responses or face-preferring responses in human infant brains. So face responses in human brains are a good topic or a good case study because they're extremely well understood in human adults. They've been studied for many decades, also in some comparative species. And human adults have cortical regions, also subcortical, but I'm going to focus mainly on cortical regions all over the brain that will show high activity in response to faces. And these include posterior visual regions like the occipital face area, which where the responses are driven by visual properties of faces, their parts. FFA, the fusiform face area, the best known cortical region, which responds to face identity, but also higher order regions like a region in superior temporal sulcus that responds to face movement and is thought to be involved in seeing people's gaze and expression. And even regions in medial prefrontal cortex thought to respond to the self-relevance and value of another person. Okay, so in adults, there's all these different regions all over cortex that respond robustly when people are looking at faces. And now we can ask, how does that set of cortical responses emerge in infancy? And what predictions can we make from these two different disciplinary perspectives? Okay, so I think one view that people have in developmental cognitive neuroscience is kind of a very strong bottom-up view of the origin of cortical function, which goes something like this. This is a caricature, but I think it's an idea in many people's minds about where the human brain comes from. So the idea is something like this. Initially, we have just very low-level representations in V1 that arise from the mapping from visual thalamus into V1 that maps kind of where shapes, contrast and edges and orientations, very low-level features. Then passive babies, human babies can't do very much when they're born, as you may know. So passive babies just lie there, and adults place their face in the middle of the infant gaze. So there's a passive infant lying there, and their parent is sticking their face in the direction of the infant gaze. Because of that, the perceptual features of faces, like they are foveated because adults put them there, they have low spatial frequency and curvy features. Those co-occur a lot in infants' early experience. And that co-occurrence in the world drives self-organization of infants' visual cortex. And then because that's happening a lot, higher order associations can accumulate. So you can accumulate first the associations that tell you when there's a face, then the ones that differentiate identities from one another, gradually the associations between face movements and environmental features. And if you do that for long enough, like probably for decades, you would get a human adult response to faces. Okay, so that's a kind of idea about the origins. Now, there's a lot of problems with this caricature, but I'm going to just start with one that's been known for ages, which is this assumption of passivity, that infants are just lying there and the world's happening to them. So that's been known to be not true for half a century now. This is a tiny example from my own lab where, if you show young babies a sample of the world while you track their gaze, so we're showing babies movies. These are 4 to 6 month old babies, and you can see that they don't choose randomly what to look at in the world. Every point of gaze, every dot is a point of gaze of a baby, and all of these babies are looking at faces and hands out of all the things they could choose to look at in these movies. Okay, so by the time infants are this old, and actually this has been studied in babies that are 1 day old. So, but this is right, here's the data for 1 day old. So 1 day old babies will choose to look at images that are face-like over ones that are not face-like. So we need an account of a baby's brain that can give it the capacity to do this, to recognize a face and to use that as input to the direction of gaze, to choose to look at faces. So somehow face recognition has to drive gaze rather than just be a consequence of gaze. Okay, so that's the small amendment, right? We need some part of the story that says, what is it about their brains that lets infants choose to look at faces? And there's a long-standing argument based on analogies, for example, to birds, that maybe there's a subcortical template that is just a very basic visual template that babies use to recognize faces, and that guides their attention. Okay, so I want to say first, like, that already means that we are adding some rich evolutionary structure into our picture of development, right? It's not just that babies start with V1 and basic features and learn the world from passive experience, but also they're born with something pretty specific that tells them what to value and where to look, right, by the time they're born. But maybe just in a few cases of subcortex here. Okay, so although I do want to say this is a cool example already where an idea about how brain development might work has to be changed in the face of behavioral data. But now I'm going to try to do this more generally, a kind of more fundamental revision of this vision of how infant brains develop. Okay, so just to look again, what is this idea that I think cognitive neuroscientists have about development, right? So they say, okay, early on when a baby's born, maybe visual features are mapped into V1 by connectivity from the thalamus. Maybe there's something extra telling you what a face shape is from a subcortical template. That leads to the slow accumulation of perceptual features of faces, leading to the self-organization of visual areas, and later, slowly, higher order associations with emotion or other things in the world would let you get richer representations. And this should be really slow, according to this view, right? So people have argued that FFA is face selective maybe by 5 years, right? So it's taking 5 years to get to the second stage here. And so in that case, then maybe the higher order associations, some people say prefrontal cortex function is taking 20 years, by some measures like myelination, for example. Hard to know what that would have to do with function. Actually, I think the question of what myelination and function have to do with one another is super interesting here, holding that aside. Okay, so here's this vision that I want to challenge. Right, and the idea is it's serial, so from back to front. Okay, and in this talk, I'm going to argue from little examples against each of these big claims. So first, I want to argue that infants are deliberately attending to faces, not just because they have the perceptual features of a face that could be detected by a template, but actually because of faces' social value and self-relevance, so this much higher order reason to look at faces. Then I'm going to argue that FFA is selective to faces in infants. So the emergence of visual selectivity is fast, not slow. And then I'm going to argue that both STS and medial prefrontal cortex respond to faces as early as FFA. And so the emergence of face responses is parallel from all the way through infancy, from as early as we can tell. Okay, so that's going to be the plan for today's talk. I'll just note briefly that we have tried a couple of times to make this argument in papers, which I'd love to hear your thoughts on if you have time to read them. In those papers, we have used evidence from many labs, certainly not only my own, but today I'm going to talk about just a few example papers from my own lab, so I have time to go into them in depth. But I just want to note here that the work the idea is based on comes from many, many labs, not just my own. Okay, so we're going to try to add some data from our group. Okay, so starting at the beginning, I want to challenge the idea that infants' experience of the world comes first passively and drives their interest in social value and self-relevance. I want to say the opposite, that infants' attention to the visual world is driven by a pre-existing interest in social value and self-relevance. And this is, right, I could use many different examples here. I'm going to use one very recent study that I think is cool. So the basic idea here is we want to study not just whether infants look at faces or not. I showed you there's been tons of evidence for decades that infants prefer to look at faces relative to things that are not faces. But here we're going to look at how infants choose which face to look at, right? If you have two faces in front of you, how do you choose who to look at? And in all of the studies that I'm going to talk about of this kind, because the dependent variable is which of two faces you look at, a face shape template could give you no help, right? If all you knew is look at faces rather than non-faces, you wouldn't know what to choose. So again, there's been lots of evidence of the kinds of features that babies use to choose which face to look at. So babies prefer to look at faces, for example, that are the same race and gender as their primary caregiver, that speak the same language as their primary caregiver, that address them in infant-directed speech. There's many features, both visual and non-visual, that infants use to choose who to look at. And here I'm going to add an even more abstract feature, which is the idea that that person is relevant to the baby. Okay, and so the way we did this experiment is we had the infants first observe an interaction between their own parent and one of the two faces. Okay, and the idea is if their own parent exhibited an affiliative behavior to one of the two characters and a neutral behavior to the other, that infants might use their known relationship to their own parent to decide to pay more attention to the face that their parent exhibited attention to. Okay, and to test this hypothesis here, we use puppet faces so that none of the other biases that I told you about can come into play. So these puppet faces don't share gender, race, or any other features with anybody the infant knows. And so the only way to choose is going to be based on the history of interaction. Okay, so here's the idea. The infants see their own parent who imitates one of the puppets.
Piku.
And then, okay, so this is custom made for each baby.
Bam.
Okay, and the idea is that the imitation is a cue that babies use to affiliation. The videos the parents made, they made without the puppets there. So the parents didn't know which puppet they were imitating. We added the puppets in by video editing. Okay, and at the test trial, both puppets are present. One voice calls the infant's name. So it looks like this.
Hi, Noe. Hi. Hi, Noe.
Okay. And the question is, which of the two puppets do the babies look towards? Okay, so this is in the first study, and this is for the example I showed you where they see their own parent imitate and then they hear their own name. And in that case, the infants look more towards the puppet that their parent imitated. So after observing their parent imitate one puppet, not the other, they then look towards that puppet. An interesting control condition is every baby also saw the videos made by another infant's parent. So this controls for whether it was the imitation per se. And they don't show this effect if it's another infant's parent doing the imitation. So they look more towards the puppet imitated by their own parent, but not to the puppet imitated by another baby's parent. Okay. So there's the yoked control data. Great. And so again, the hypothesis here is that infants infer that the new character imitated by their own parent is relevant to them. Okay, I'm gonna just go, right. So we wanted to make sure this was specifically about self-relevance. We did a version without the name, right? So this is an independent replication with the name calling, and then a control condition when there's just music playing and infants look specifically during the name calling. But the actual control condition that I think is the coolest here is in this. So here's another version of this experiment where one infant calls the infant's own name. Okay, so here's
Hi,
the same
Noe.
thing I showed you
Hi.
before.
Hi, Noe.
And in the other condition, they call a different baby's
Hi,
name.
Harper. Hi. Hi, Harper. Hi.
Okay, and so here's the replication again, where when the puppets called their own name, the babies looked towards the puppet imitated by their parent. When it was another parent, they didn't. And when their own parent imitated one of the two puppets, but then the puppets called a different baby's name, they also didn't look. Okay, so what I want you to take away from this data is that preverbal infants' attention to faces is not driven by only low-level perceptual features like that's a face. In this case, it's also driven by a very abstract idea that this character is relevant to me right now. That is, they were imitated by my parent and they're calling my name. And those non-visual features, right, the history of the interactions with their parents, what the voice is saying, those non-visual contextual features are driving infants' choice to look at one face more than another. Okay, so that's the specific example from our lab. This is actually recently published. But again, the bigger picture that I want to give you here is that there's now tons of evidence that infants attend to faces' social value and self-relevance very early on in development. And so whatever story we tell about infants' brains, it needs them to have the capacity to recognize a face and also to bind it to event histories, to auditory cues, to all kinds of other features, and then to drive goal-directed looking, right, to deliberately looking by very early on in infancy, right? We can't have an account whereby these multimodal associations or event histories get bound to faces gradually over development because they're driving babies looking very early on. Okay, so that is the first claim. So how does that relate to development? Okay, so now I'm going to go to neuroimaging data. And as I said, there's been a long history of people trying to ask, when do visual responses to faces arise in human development? And a popular and compelling argument that this occurs gradually through self-organization of bottom-up input from visual areas. So the idea here is that, infants start with low-level features in vision, like retinotopy, spatial frequency, and curvilinearity, the features that govern early visual representations. Through pre-existing connectivity from visual thalamus, these are mapped into into mid-level visual cortex. And then it's the co-occurrence of features in the environment that causes, for example, responses to scenes to occur where input is from high spatial frequency and peripheral sources, and to faces to arise in places where the input is dominated by low spatial frequency and curvilinear input, right? So this is a very bottom-up account of a gradual emergence of responses to faces. So that is a compelling theoretical idea, but to test it, it would be great to directly measure the origins of face responses in human infants. So we set out to do this by measuring responses to faces in awake human infants using fMRI. It needs to be fMRI rather than NIRS or EEG because we're asking hypotheses about the specific location, right? Is it in FFA as opposed to in other locations? And because the FFA is deep, it can't be observed by NIRS. So we needed to be doing fMRI, and it needed to be in awake infants because we want to measure their response to visual input. So I'm saying all this because at the time, and many groups were trying to scan human infants, but it was common sense that you can only scan them asleep. So I don't know how many of you have followed for the Human Connectome Project, for example, or many projects attempting to study infants using MRI, but almost all of them study sleeping babies because how are you going to get a human baby to stay still? Okay, so that is true. It's crazy hard. Okay, so we developed a series of special coils. This was the first generation infant coil. That they're designed to be like a car seat. They have a high density array right around the baby's head. You can see the frontal pedal. And then we sent babies into the MRI machine with an adult to look after them to make sure that they were okay. And then the babies could watch a movie through a mirror that was over their face and looked at a screen behind them. And this is a representative baby, so you can see how still they are during the scans. Okay, so this was our first attempt. We scanned 17 babies over the course of 3 years. I'm just giving you a sense of how much time they were in the scanner. So the gray bars is how long the baby was in the scanner. And then we scrubbed motion contaminated images from the data we had collected, and the orange is what was left. So there's, as you can see, much less data left after motion scrubbing, just to give you a sense of what a baby's brain looks like in the MRI. I mean, I hope you can tell there, like these images are utterly useless, right? The baby is moving so much, the distortions are absolutely massive. And also, we had, for this experiment, used a quiet sequence because we wanted to be extra careful about the baby's hearing. And this quiet sequence also introduced distortions. So these data were hard to work with, I will just say. Having said that, we managed to get some results in 9 babies. As you see, there were 9 babies that had some non-motion contaminated data. Looking at both dynamic scenes and dynamic faces. And in these babies, we could see the first hints that the spatial organization of responses to faces and scenes in mid-level cortex of infants, these were on average 6-month-old infants, looks a lot like adults. So this is the scene responses in PPA and the face responses in FFA visible here. And in functional regions of interest analysis, where we independently select the voxels with some of the data and measure the response in left out data, we could confirm that indeed there are parts of mid-level infant cortex responding to faces more than scenes and to scenes more than faces. Okay, so that was our first attempt. Could we do better? I will say that data quality was a major challenge. You can see there are some babies in whom we got a reasonable amount of data. Those are my first child, my second child, and my postdoc's child, which is not a sustainable way to do this research program. So the next question was, okay, what about a broader sample of the human population? And for all of the babies for whom the parent was not me or my postdoc, we were collecting on average 5 minutes of useful data. So could we do better than that? So my next brave graduate student, Heather Kosikowski, spent her PhD, she's just graduated a month ago, scanning a lot more babies. She amazingly recruited 86 babies, of whom 52 had some usable data. This baby number 54, who was in the scanner for days and never gave us any usable data, was a particular tragedy. But anyway, you can see that many, many babies did eventually give us data. Also, to combat the distortions from our earlier coils and protocols, she designed a next generation infant head coil where instead of the frontal panel coming down like this, it slides over the top and slides over special infant headphones that are better at hearing protection, so we can use better, higher resolution protocols. And this helped. So you can see in our original coil how distorted the functional data were. This is comparable data from the new coil. So we're excited that that seems to really have helped improve the resolution for the same amount of data. Okay, and so that led us then, both by having much more data, way more babies, so much more data in that sense, but also better quality data per baby, start to ask about the origins of phase selective responses. And so in this study, the babies all saw 4 conditions of videos. So all the stimuli are dynamic videos of faces, body parts, object scenes, and a curvy, wavy baseline. Oh yeah, here you go. That's what they look. That's an example of what they look like. And we could use then individual functional regions of interest to identify these regions and then look at their responses. So for example, in 26 infants scanned in the first half of the data collection, this is the FFA, which you can see now is clearly highly selective to faces in these infants. Yeah, here we go. So this is in a group of infants aged 2 to 9 months. The FFA is highly selective. Okay. And one thing that we can do, because we have all 4 conditions, is ask, what is the best prediction of this response? Is it the faces per se, or is it features like curvilinearity and spatial frequency and so on? So we modeled, for example, the low spatial frequency content of all of our conditions and the curvilinearity in all of the conditions. And you can see that the faces are not, the faces in this stimulus set aren't more, don't have more low spatial frequency content or more curvilinear input, but still do drive the FFA more than the other 4 conditions. Okay, so here the argument is, okay, by this sample, so by age 2 to 9 months, the FFA, which I'm showing you here, responds selectively to faces. So it's not responding just to low-level visual features. And so going back to our bigger picture argument, my argument now is that at least what I've shown you for the FFA is that the emergence of selectivity to meaningful stimuli happens fast in development. It doesn't take years because it's there in this group of 2 to 5 month old infants. Okay. So, and now the question is, okay, how can we argue about whether it got there by the accumulation of bottom-up input or by including top-down input from other regions? Okay, so one way we can look at it is by asking about, okay, yeah, so this is now combining data across both coils for more power, just to show you what it looks like when we add. So this is now 39 infants. This lets us now compare the ventral temporal regions, which includes FFA, to the higher order regions and ask, okay, if it's true that higher order associations accumulate slowly from back to front, then what we should see is that although the FFA is already phase selective in infants, STS or medial prefrontal cortex might not be. Okay, and so one possibility for these higher order areas is that either or both might actually have no phase selective response in infants at all, right? So certainly for medial prefrontal cortex, right, we've known for a very long time that it's very undermyelinated in human infants. And so it might be that the bottom-up input needed to drive face selective responses just hasn't arisen at all yet in this infant sample. So the first question we can ask is, okay, is there any response in these regions to faces more than to the other conditions? Or the alternative is, well, maybe 2 to 9 months is a very large age range. That's a quadrupling of infants' visual experience. And so maybe what we'll see in these higher order brain regions is a gradual increase, so that the response to faces will be present only in the older infants. Okay. And then the third possibility is that we might find it's conditionally. So only the babies who already have a face response in FFA already have a face response in higher order areas. So those are the three ideas that we're going to test here. The first question is a selective response to faces present or absent in these higher order areas, superior temporal sulcus and medial prefrontal cortex? And so here's superior temporal sulcus. And this was replicated independently both in the old coil with all the distortions and in the new coil with the much better resolution data. And then the same thing in medial prefrontal cortex, highly selective response in the full sample, which we could see in both the old coil and the new coil. Okay, so it's certainly not the case that face selective responses are absent in infants in these higher order areas. But what about developmental change with age? So again, for each of these regions, we measured the response to faces relative to the other conditions. But now we did it for each individual baby in whom we could collect any data and asked whether that changed with age. So I'm going to show you here in a small inset plot for the STS and the medial prefrontal cortex, the change with age. And we actually saw no evidence of change with age in either region. So to the degree that we could measure face selective responses in these babies, it did not seem to be different for the younger babies or the older babies. And we could look at that more formally by looking specifically at the youngest infants, so just the subsample of infants who were between 2.5 and 4.5 months old. When we looked at those babies, the response in, oops, this is disappearing. Okay, here they all are. So the response in FFA is actually now marginal, but it's still very clear that there's a face selective response in both STS and medial prefrontal cortex, even in babies, all of whom are less than 5 months old. Okay. So it's clearly not the case that it takes years for these higher order areas to become face selective, because here they're face selective in 2.5 to 4.5 month old babies. And then the last thing we looked at was this idea of conditional dependence. So is it true that only babies who already have a face selective FFA could also have face selective responses in STS or MPFC? We did this by subsampling out of our full sample of babies, subsets of 15 infants, 2,500 times. And so each time we asked whether each of these three regions showed a face selective response. And the idea is if there's a back to front development, then what you might see, the prediction would be that there would be many subsets that had a response in FFA and ventrotemporal cortex, but not yet in STS or medial prefrontal cortex. And then the babies that had STS responses would be a subset of the ones that had the temporal responses, and the ones that had medial prefrontal cortex responses would be a subset of those. So we're going to use a Venn diagram to ask, are the babies that show responses in the higher order areas a subset of the babies that show face selective responses in the visual areas? And we found no evidence of that at all. So the red here is the samples in which we saw babies that had only ventrotemporal cortex, not the other two regions. And you can see that is not a good way to represent these data at all. So the most common pattern we found was that babies had all three regions, but the second most common was actually that the face response was present in medial prefrontal cortex and STS and not present in FFA. So no evidence at all that the visual regions were preconditions for face responses in the higher order areas. Okay, so what I want to argue from the neuroimaging data, the fMRI data, is that the emergence of face responses in infant cortex is fast and parallel. So it's not emerging gradually, and it's not emerging in bottom-up sequence. It is emerging fast and in parallel. Now, one totally interesting possibility is it's not just parallel, but it's actually top-down, right? I just showed you that a bunch of babies seem to have responses in the higher order areas to faces and not yet have responses in FFA. So I think that's a really interesting idea, whether that could be true and how we would understand it. And I'm just going to give you one hint of one way that that might work by coming back to the role of social value in the biasing of infant attention. To do this, I'm going to show you work from NIRS, near-infrared spectroscopy. I don't know if everybody here knows about NIRS, so just in case, this is a video of a baby putting on a NIRS cap. This uses optical measurements to measure signals that are very similar to the bold signal in fMRI. So it's measuring blood oxygenation through an optical measurement, and it can only measure it on the surface. Okay, so in this experiment that I'm going to tell you about, we measured medial prefrontal activity when babies were facing the kind of choice I described at the beginning of my talk. So there were two faces, and babies needed to decide which one to look at. Here again, of course, since it's two faces, a subcortical face shape template can't help you. And here we gave babies an auditory cue to which face was more relevant to them. So one of the faces, here we use human faces, one of them talked to the babies in infant-directed speech, and the other one spoke neutrally. And then we measured activity in their medial prefrontal cortex while they were watching those two faces, and afterwards measured their attention to the two faces. Okay, so here's what this looked like. One of the faces, this is the neutral face. Okay, so one person speaks to the babies in a neutral way. It's all nonsense syllables.
Gesi fawo namu, mufa nasi gewo.
Okay, and so the other one speaks to the baby in an infant relevant way, infant directed speech. And then, and we're recording mirrors while they're watching these two faces. Then afterwards, we remove the mirrors and we give them just the two characters, both silent on the screen. So they just see the two characters, both smiling, both silent. And the question is, where do the babies look now? So they have to, if they're going to make it, they have to remember which of those two faces was associated with a voice talking to them in an infant-directed way. Okay, and so what I'm going to show you here on the y-axis is how strong a speaker preference the baby showed. That is so 0 here is equal. If they're above 0, they showed a preference to look at the person who previously spoke to them in that infant-directed way. Which many babies do, right? So more of the babies are going to be positive here, but predicted by, while they were watching those two speakers, how much activity was in medial prefrontal cortex. So the more medial prefrontal cortex was more active for the infant-directed speaker, the higher up the babies are going to be on the x-axis. Okay, so here was one study example showing that the more medial prefrontal cortex responded to the infant-directed speaker during the original exposure, the more the babies looked to that speaker later. Okay, so again, and this is in 6-month-old babies. So by 6 months, the more that an infant's medial prefrontal cortex is responding to the social value of a face, the more the baby looks at that face, right? So the more visual input from that face the baby will get. And so again, going back to the big picture synthesis here, I want to suggest that, like adults, pre-verbal babies are responding to faces in terms of their visual features, but also in terms of social perception and social evaluation features, like their self-relevance and value to the babies. And these higher order cortical regions are part of the process that drives the input to the visual regions, either exogenously by driving what babies choose to look at, and possibly even endogenously by directly giving top-down input to visual regions during infant development. Right. So infants see what they're looking for. They look towards parts of the world that have social meaning and self-relevance. These seem to be encoded by the same regions in infants as in adults and present from very early in infancy, and lead to a bias in the perceptual input that infants receive. Okay, so, and the big picture idea here is that social representations and motivations arise very early in and influence the development of human infant brains. And I think thinking of brain development this way poses different questions for the sources of cortical development, and actually the role of connectivity, and maybe even the kinds of cross-species studies that we should be doing, just to connect it back to the workshop. Okay, so thank you very much to the babies and their families, to my lab, and to our funders for this research. Thanks.